you can actually record videos in the eye of an insect, right? It can say, this is what the insect saw at this moment. And so you have essentially a digital camcorder that's entirely biological.
Okay, so welcome to the Biohack podcast powered by Bronic. Here's what the heck security means for biology. We interview some of the world's mad scientists and rad hackers across deceivingly unrelated disciplines to uncover what the future holds and how we can design against crime. My name is Dr. Miriam El-Gabri, your host, and today we're speaking with Professor George Church, professor of genetics at Harvard Medical School, as well as at MIT, but he wears many, many hats, including leading synthetic biology at the Wise Institute and much more. Welcome, George.
Great to be here. Thank you.
I'm super excited to have you, but before we begin, I'd love for you to tell us a little bit about your background today.
Oh yeah, so this is my pond right outside my door. A security camera fell into the water a couple of years ago, and it still works. And we have 20 baby koi that made it through the winter, and we have 4 adults that are almost 3 decades old now. Yeah.
Amazing, amazing. It's truly magical. I'm kind of jealous, and yeah, you totally win on the background today. I love aquariums. I find peace at aquariums.
Yeah, they're I find this very peaceful.
Yeah. So George, I mean, there are a lot of adjectives to describe you, legend being one. I think the most common one, at least in the UK and our labs at UCL, is the god of synthetic biology. You have co-authored more than, I think the last number that I calculated here is 650 papers. You have more than 150 patents, and you either co-founded or advise more than 50 companies. So you're really redefining what we mean by serial entrepreneur. Usually that's 2 to 3 companies, George. What's happening? Can you walk us through a day in your life?
Right. Well, it's actually pretty simple at this point, but it took some effort to get here. And you know, I have co-founded 46 companies. Most of them recently have been with postdoctoral fellows and a couple of graduate students that they want to take their things that we've invented together at Harvard or MIT and move them into the real world, and they're very highly motivated and excited about that. And I think that has worked better than many other business startup mechanisms that I've tried in the past. And they like each other. I mean, it's a high priority, both my lab and my startups. They there's not this cutthroat competition. They support each other out there in the outside world. Anyway, yeah, it's a day in the life is basically I eat once a day, and I spend a lot of time with my lab and a little bit of time with my postdocs that are now out in the world. And then even smaller time now for traveling. I travel mostly by internet, which is way faster and more efficient. Yeah.
Ooh, yeah. I want us to talk about that, because I still recall our last conversation regarding the benefits of calls on the internet versus in person. But before we do that, I mean, just to say that you're really not a typical academic, and you alluded to this just now in terms of what a successful kind of startup environment can be. Can you just talk us through your mental model and how it is that you're really combining a lot of converging technologies as well with your work in your startups? And feel free to kind of share your favorite startup too, if you're allowed, out of the 46.
Yeah, okay. So one of the things that makes us a little different, it starts in how we recruit people into the lab. There's there's a lot of self-evaluation, self-recruitment where they see what we're doing from a long way away and either apply to Harvard, MIT, or postdoc. And that is a kind of a positive feedback loop that keeps, evolving in a positive way. And they come in thinking that they're going to do some really basics research that also has some societal implications and maybe some, order of magnitude technological implications as well. And if we can get all three of those, then we know we got a good project. But they come in with that expectation, and so it's not surprising that by the time they're done, they have an idea for a startup. So in the old days, it might be that going in industry was like second best, and very often, if you were working for a big company, it was second best. You had no independence. Had very little say. But when you start your own company and everybody starting it is basically C-level, CEO, et cetera, then it feels a lot more self-directed, and that plus the fact you invented it creates very high motivation.
Absolutely. And one of the things that I noticed when I first, when we had our very first conversation, is your approach to what others might say are radical ideas. I found myself while speaking to you and kind of sharing what little crazy idea I had, that you tended to flatter each kind of possible dichotomy rather than kind of come in there with the limitations. So I can totally see that also adds to the motivating factor and that positive feedback.
We're always looking for win-wins rather than trade-offs or contradictions. Looking for the win-win. We're also tend to accept millions of failures for every success rather than like trying to create the perfect product, the perfect company from day one. And that's partly inspired by, I think, one of the things we help pioneer, which is libraries, molecular libraries. So in a molecular library, you're looking for that needle in a haystack that's really very valuable to society. And it doesn't matter how many hay pieces there are in there. So failure is an option. In contrast, that's our motto rather than the NASA model. Although we're aiming for kind of similar engineering goals.
Yeah. So take notes, everyone, to find successful companies. Okay, let's talk to us about the internet versus in-person perspective that you have.
Well, it's something I was advocating long before COVID, but nobody really wanted to admit that it existed as an option. It was always, that won't work. Video conferencing doesn't work. But it has the advantages of, it's much more carbon friendly. Your footprint is smaller. In terms of timing, I spend a fair amount of time in conversations like this one, and when I'm in the air, typically I'm flying economy like I should, and there's just no practical way that I can have this conversation in a 48-hour round trip to Asia, and also just wear and tear on the body. You tend to get a lot more back pain, risk of deep vein thrombosis and so forth, not to mention infectious diseases in that travel. So it just has numerous advantages, and also because with the time that's freed up, getting rid of that 48-hour commute, I can now do 48 one-hour video conferences with places I wouldn't normally go, like Uzbekistan and Kazakhstan and kindergarten classes and things that those are actual examples from the recent past that I just wouldn't have considered before.
Yeah, absolutely. And I'm also going to share what should I call it? Should I call it not a prank, but like there was like a mysterious presence where you were at some BioBeta conference. What was it, last year or a couple of years ago? But you weren't, in the sense that you were there speaking and people heard you speak. But when people, you weren't there physically.
Right. Yeah. There's quite a few of those. In fact, I'm getting more and more invitations that don't even ask whether I want to show up or not. They just say, look, we got a virtual session. You want to be in it, right? You know?
Yeah.
Yeah. And there's a lot of panels I've been on where a lot of the people are really there in meat space. And I'm like this hovering, you know, Wizard of Oz, gigantic head. Not my choice, but that's how it looks. Yeah.
Yeah. Okay, one step further. What is it? I mean, there are like various technologies now coming into kind of media, including like metaverse and like all these into like Web3, etc. I mean, how do you envision art communication like this in a podcast in a couple years? Are there going to be like haptics? Because you know the common thing people say, yeah, but what about their benefits and being in person, like reading body language or like other things that get lost in this like
Right.
video kind of recording. What do you have to say to that?
Yeah, I'm a big fan of body language. Not a big fan of like shaky hands, but yeah, I don't think there's any reason why we couldn't convey body language. We've chosen to focus on the face, and this is another social revolution. We don't know where it's going, but it could be that we decide that the facial cues are more important, and we develop them in some way, or maybe we have a little cutout over here, which is what the rest of my body is doing.
To read the rest of the body language.
But one thing that's really nice is, you know, when I used to go to like boardrooms, you know, or you know, meetings where you have this long table, you really can't see the people to your right and left. You can just see them right across the table. It's even hard to see across the table if the table's long enough far out. But now, you know, you can fit 25 people and you can actually catch, you know, the nuances of all their faces because they're all looking right at you. They all have their name underneath them. So, you know, so if you hear somebody's name and you don't know how to spell it, well, you just look at their little name tag. So I think there's just, from a social interaction standpoint, it has a lot of advantages, especially, you know, if you're not planning on being intimate, let's say, if you're just trying to get some business done.
Yeah. Why do you say that you're not a fan of handshakes? Is it like a biosecurity?
Well, first of all, it's not a universal human trait. It does spread contagion. Its source of you know, historical was that it showed that you weren't carrying a gun or a knife. But I don't think, you know, first of all, I don't think it's accurate, and it's anachronistic. But you know, I think a bow is appropriate. You know, asking about how the person and their organization, their family is doing is more appropriate than a handshake if you're going to do something ritualistic.
Speaking to the kind of the mission of your research, here I have the sentence actually of how you describe it: transformative technologies for reading and writing three-dimensional and four-dimensional structures at any scale, inspired by but not limited by biology. And what I specifically and particularly really like is that you don't end the sentence there. You say, with attention to ethics, safety, security, equitable costs, and communication. And for people that haven't visited this part of your website, there's more than 30 publications and videos and press releases focused on this latter part, which is like the security and safety. So what does cyber biosecurity or security mean to you? And I say "or" because sometimes with these terms, there's a bit of controversy, and we can totally go into that as well.
Yeah, so sometimes ethics is some abstract thing, but in this case, I'm talking about the concrete ethics of accessibility, worldwide accessibility, so that anybody who wants it can get it. And there are very few technologies that have actually reached that level. I think one of the few is like smallpox. That's extinct, and so it's distributed to everybody, doesn't need to be updated and upgraded. Okay, so that's one part. The safety component is almost every form of engineering has a safety component. Now, synthetic biology being one of the most recent new categories of engineering hasn't had quite as much time to develop it, but it's been there from the beginning. Potentially for good reasons. And so every time we develop a new technology, we immediately ask, before we even publish it, is there a safety component that we should champion first or simultaneously? So for example, when we did gene drives, our first 3 papers were on how you keep them limited, how you reverse them, how do you keep them on the island away from mainland. Those were the first 3 papers. And when we improved DNA synthesis by about 4,000-fold, we said, oh, this suddenly is enabling for, say, synthesizing viruses. We should have, so I produced a white paper in 2004 that talked about how you can track the chemicals and the know-how and the devices and so forth that's necessary, because there's a limited number of choke points that were available in 2004, and it's still true. And in fact, we're moving to a place where we might have 100% of synthetic DNA that's commercially available anyway, whether it's desktop or via mail, to be checked by software that checks for things that you shouldn't accidentally or intentionally be making.
Is that the one of the I think it's referred to as a double check, right, for DNA synthesis companies? Is it just a check across a list of pathogens, or is it and then who is also placing the order, or is it something more?
It's select agents, generally toxins and pathogens of all shapes and sizes. It's aiming for smaller and smaller pieces of DNA. So when the first laws were passed, partly I think due to my white paper, they were like 200 base pair double-strand pieces, but we can now detect much shorter examples. Also, if they spread the order out over multiple companies, that should be coalesced and compared, because that in itself is alarming if somebody spreads an order out.
Yes, which was a huge concern actually in terms of the 200 base per 200 base pairs order for the sequencing. So okay, let's dive deeper than that. With the increasing use of these gene editing and other biotechnologies, what other kind of new security threats are we facing today, and how can we develop strategies to address these threats?
Well, some of the ones that I addressed when I was starting the Personal Genome Project, as the third example here, was threats. In principle, you can now sequence anybody's genome for $250, even if they just leave their DNA around in their environment on coffee cups or dandruff on the desk or whatever. Kind of like Gattaca, except certain
I was just gonna say
surreptitiously. So that's an issue. And furthermore, once they have your genome, they can make a copy of you or your gametes, and then do paternity suits or crime scene hacking. And so I think these are issues that need to be addressed. In the Personal Genome Project, we specifically recruited people. We were I think the only organization, maybe still, that was 2005, even today, that consents people fully informs them about the possibility that their DNA can get out electronically, can already get out biologically, and that it might be used commercially. So most material transfer of cell lines and information don't really take those into account properly when they do they either pretend they don't exist or they just forget about them or something.
I think the best example that I use for this type of thread is I don't know if you've seen Heather Dewey Hagborg's art. I think it was around, was it 2011?
Oh, is this from cigarettes?
Yes, where she collected genetic material from cigarettes, and then
the DNA that you leave on discarded food or cigarettes is enough to reconstruct your face. Now that is based on science from I think Penn State University initially, and it is remarkable. I mean, if you look at identical twins, right, there is no doubt that their face is encoded in their DNA. I mean, it's just identical twins look exactly like each other typically, and they look quite different from all their other siblings.
Yeah, I wonder in terms of the algorithm that she used, has it improved in the conversion from like the
It keeps improving, and it will continue to improve, no doubt. But you know that it's one of these things that's highly heritable, so you can eventually get it, better and better. Other things that are low heritability, say Parkinson's or something, and there you have pretty good prediction of the small fraction that is heritable. So, but faces, I think, is really one of the most lovely demonstrations of just how heritable things can be. I think we like to say that it's genes plus environment. I had one of the few meetings that was called Genes, Environments, and Traits, and but in this case, unless you get severely burned or some disfiguring infectious disease, you're going to look like your identical twin.
Mm. To go back to one of the questions that I don't think you've answered directly, what does cyber biosecurity mean to you?
Oh, cyber biosecurity. So I partially addressed it by this personal genome project where your cyber version of your genome could get out, but because there are, thousands of examples of hacking of biomedical information, hospital information, so forth, so that you shouldn't have false claims about your security. But there's other which is that if you have information on on synthetic biology, right, this can be even more significant than personal information. With synthetic biology, you might have things that are proprietary information, things that are of national security, and so forth, and they can be hacked just like anything else. But then it's relatively easy to make them. So if you hack information on, let's say, a aircraft carrier, it's going to take a while before you can build a atomic aircraft carrier. But if you hack information on plasmid, you just order it, right? And a few days later, you got it. So, but I don't know if that addresses what you were interested in, or.
No, I think that's what's another really good example to show this interplay between cyber and bio that we have, even with COVID. Who is it? I think it's Professor Jurendi that mentions the Switzerland example, right? How did COVID first arrive to Switzerland before the first actual case was recorded at hospitals? And he uses that example to show that COVID was actually synthesized, it was sent via the internet and synthesized first in the lab, which makes it prior to arrive in Switzerland prior to the first case recorded, which is kind of.
But it wasn't really a case, though.
It wasn't exactly. So if you
it was an arrival, yeah. Okay.
Yeah. Mm-hmm. But it just shows that interplay and exchange of the physical and the kind of the digital version of something like a pathogen.
Yeah, that's true.
Perfect. So how do you envision the role of security or cybersecurity in biology and biotechnology evolving over, let's say, the next decade? What kinds of new challenges and opportunities do you see emerging in this area? And maybe should we just be a little more fun and kind of change that question and say it a different way? If you were to design a cyber biosecurity survival kit for for a dystopian or even utopian future, what items would you include? Let's be a little more fun.
Yeah, well, so if you want to protect your own DNA from hacking, there are articles written. I think the first one was in The Atlantic about how you can make personalized weapons, things that can, where you don't have to be in the room and you don't have to, aim a sniper rifle at somebody. It's just you've got a microorganism with their name on it, and it attacks them. So from that standpoint, if you want to protect your own genome so that they don't have that, then you really have to wear a full body suit or not go out. Now, we have a little bit of practice of not going out recently, but I don't think there are not that many people that enjoyed it, but it is possible.
I also liked, wasn't there, in our previous chat, we were talking about the possibility, or this is, I think, in your, for everyone who doesn't know, George Church also has like a list of ideas that you aren't or aren't necessarily pursuing. And it's for people, it's kind of like a crowdsourcing, right? If someone's interested, they can contact you, and then we can paste that all of these links in the podcast. But one of them, which we had discussed, which is kind of relevant in this topic, if you're happy to talk more about, is like the biosecure domes or homes. Yeah.
Right. So this has a number of origins that intersect. One of them is if we're going to build colonies on space, which we certainly should do, because there are all kinds of catastrophic things that could happen to Earth that we can't prevent. So we should have a backup. But if we're going to have that colony, we need to debug it, and debugging it on Mars, which is, involves 2-year cycles, is awkward. If something goes wrong, you're many months, if not years away from the nearest emergency room. But the other aspect of it, if there is a catastrophic event, kind of global catastrophic event, and you don't yet have a colony, or you yourself are not on that colony, then you probably want to have one of these biosecure homes. And it can either be built from scratch, or the white paper I have describes how to retrofit a normal home. And the key components are complete recycling, which we've never done with humans, but we've done with many other animals. Also, protection from electromagnetic pulses, which, and solar flares, and asteroids, nuclear winter, and so forth. So you need a source of energy that is resistant to, loss of sunlight. So geothermal is a good example of that. Anyway, so the key component, I think, at least for starting up, is having some source of food which you can generate from electricity, where everything is recycled, and you can seal your house in such, and the supply chains are no longer necessary. So supply chains are going to be one of the first things that goes with most of these global catastrophic risks, and you want to be independent. And so if you have just some source of energy, and that's renewable, then you can make your own food and probably other materials and devices. And it's an interesting challenge for synthetic biology, is to make this, universal synthesizer, but starting with food, I think.
So what would that look like? Are we talking about like algae? Because I'm just thinking they photosynthesize.
An example, if you want to, all this is revolutionary, so it's not going to look the way things. But if you want to map it on the things that exist today, it would be something like spirulina, which is an amazing cyanobacteria that grows at very high pH, up to a pH 11. It's like one of the highest pHs of any organism. And so there's very few predators and parasites that can mess up its life or its productivity. And all you have to do is add certain wavelengths of light. So you can take the broad spectrum light for solar power and remap it into the very efficiently into the right wavelengths. So you can actually end up with higher photosynthetic efficiency than growing it outside. And of course, if your source of light is geothermal, then it's even more important to do that remapping. And it has a broad, then we have the whole spectrum of synthetic biology and chemistry that can transform it into any taste and texture that you want, a whole variety of materials, including lots of inorganic materials. So the number of inorganic, I just recently wrote a paper in astrobiology where I listed a good almost all of the known inorganic chemistry that happens in biological systems, and it's it's not very limited.
Oh. So I'm picturing these domes and self-sustaining
Well, it's your home that you seal
yeah.
initially. I mean, you can make you can make a dome like it if you want, but the main
Yeah,
thing
I
is
keep saying dome maybe because
your
I'm personally
kitchen, you have essentially a food manufacturing system as a starting point.
Yes. So does that look like kind of the dream for any synthetic biologist or scientist, the democratization of practicing synthetic biology as if it was just like cooking in the kitchen? Is that what it looks like? Or?
It's like the combination of cooking and gardening, right?
Yes.
All in one. Yeah. And you'll share recipes, but the recipes will, start from photons or electrons and go all the way to, fancy cakes, that are good for you.
Yes. I mean, that's awesome. I want this to happen now.
Yeah, me too.
Yeah. I mean, how do we start something like this? And I know synthetic biology, and I think it's now in the UK at least, it's being rebranded to engineered biology because, of course, it's had its ups and downs, especially in terms of how the public perceives it. And at least personally, I came out of my PhD with a bigger question, which is kind of how do we socialize things like biosecurity and security and science really to involve public participation more and upskill. But yeah, can you talk to us a little bit about kind of how you see these aspects, what you think about democratizing biology and synthetic biology, and what the challenges are in kind of getting to that point where we have domes and we're quote unquote cooking, but actually engineering our own food.
Yeah, I like the term engineered biology. The problem with bioengineering is it typically is talking about artificial limbs and things like that. And the problem with genetic engineering is that was kind of co-opted for like single gene molecular biology. But engineering biology is much broader. Increasingly, we're engineering developmental biology, multicellular systems, like making pig organs for transplants and engineering, agriculture and engineering the Arctic to be a carbon sink via cold resistant elephants and all these kind of big ecosystem and developmental biology. So biology is synthetic. It just keeps conjuring up synthetic chemistry, I think. So, and all the problems that came with it. While biology conjures up, breeding cattle and pet cats and dogs that are very different from their ancestors, and all kinds of flowers and foods. Yeah, those have been mostly positive experiences, and it's a tiny step from engineering your domesticated plants and animals to something that works in your kitchen to make just about anything. Actually, I teach a course called How to Grow Almost Anything, which is at both in person, MIT and Harvard, but virtually in 400 places all over the world, along with David Kong and Joe Jacobson. We've been teaching this for many years. And that's what I'm trying to live up to. How do we grow almost anything in everybody's home in such a way that it's more equitably distributed than if you had to, go out and buy fancy things you can't make at home.
And this course that you just mentioned, how to grow almost everything, is that so? Can anyone kind of virtually join in?
I believe that it's unlimited virtually. There may be some limit. You need to have an internet connection at a minimum, but there may be other limitations. But it's intended to be for everyone. Yeah. And we have labs. We have labs that during COVID were virtual, so we had like a combination of robots and postdocs that would help keep the lab running, so that students could program the robots distantly and have it make things. And so that's getting again close to this. What what you might do at home. It's better than a 3D printer because 3D printers can't typically have trouble making things that are at the nanoscale and at the meter scale, which biology has no problem making both of those.
Yeah, I'm actually still very eagerly waiting for progress in bioprinting, 3D bioprinting. Do you see that reaching a point where we can just print on the go, like biosensors or like cell-free sensors? Or I know we're not. I think we're not there yet with.
We're pretty close. We're actually quite close. We collaborated with Jennifer Lewis's lab at the Wyss Institute at Harvard, and published some on taking essentially inkjet printing cells. But you can also harness developmental biology or various combinations of these two, and developmental biology is amazing because when you try to say inkjet print anything in 3D, as you try to scale it down in resolution or higher resolution, it say tenfold. So you go from say 0.5 millimeter down to 50 microns or even 5 microns or less. That tenfold increase in resolution costs you 1,000-fold in print time. And the same thing if you want to scale it up. If you want to scale it up to the size of, say a forest or something like that, a factor of 10 scaling up in size is another factor of 1,000. So if you scale it down in resolution and up in size, then you've got just tenfold. That's 1,000,000-fold. Okay, so it doesn't scale gracefully, but biology, engineered biology, or even regular biology, is atomically precise. So you can't really scale much further down than that, and and it handles ecosystems, and it does it essentially for free. I mean, there are versions of it that are expensive, but there are versions of it that are free. Most of the world's biosphere was initially free.
So, okay, so that, I guess that's why there's a preference to engineering biology than like I guess trying to scale the machinery that we currently have. What does the Arduino type model look like for biology then? For people who are familiar with Arduino and can kind of understand that like plug and play concept for electronics.
Yeah, so I built some Arduino circuits, and it's probably the closest analogy is the iGEM BioBricks kind of way of thinking, where you have these modules, which you could start out, the simplest module is say a gene, DNA, RNA, or protein, and then you make them interoperable, so the output of one fits nicely with the other. And so that's the engineering part of engineering biology, is really making those interoperable parts, manufacturability and so forth. But that isn't the only way to do it. And in fact, I think what's both the bioprinting, Cartesian coordinates, and the computer-inspired modules, or the Arduino-inspired modules, is us forcing our long and wonderful history of inorganic engineering onto biology. But the other way is to let biology tell us what it could do. So, for example, if I can take a really complicated object that I've made, however you made it, and let's say it's a cubic meter with atomic precision. Okay, that would be almost impossible to make today by any method other than biology. But in biology, you can make a copy of that in as little as 15 minutes. Or the absolute record for doubling time is 15 minutes. A lot of things will take longer than that. But the point is, developmental biology can make copies of worms and flies in days, essentially doubling time of 40 minutes, in that sort of range. And what it is, you don't program it by Cartesian coordinates. You program it in a language we don't fully understand yet, but it allows you to make decisions about branching, about migration, about electric and chemical gradients, and so forth. And if you speak in that language, then you can get these incredible doubling times where you can, imagine taking a farm and making a copy of it in 15 minutes, something like that. It's if the farm were made out of Vibrio nitrogens, it would be easy, and you have enough food or sunlight.
I only recently actually learned about I think you were referring to also like a bioelectricity, right? Where it's this like cellular level control of the development of an organism, where you're not tweaking the genetics, let's say, to guide its development, but you're tapping into the electrical or cellular level to change or guide its development. I think a really good example is that the worm, right, where it was like cut in half and it still knew how to grow ahead, even though.
Yeah, so I didn't mean to single that one out. I mean, there's a whole variety of different things. It's like cells will chemotax. They'll have cell adhesion things. They'll have chemical gradients. They'll have, but the point is we need to learn all these basic principles in order to program our 3D printer. So like right now, 3D printers are mostly Cartesian, and they either involve, inkjet deposition or molten plastic deposition or subtraction or powder dust that you sinter, so forth. But there's a whole nother set of basic building components that we need to master. I think bioelectricity is a minor one, but but you probably need to master all of these things.
Yeah. No, that's really cool, and I can't help myself actually, but think of I don't know why. Maybe you can help me understand why. But when you say chemotaxis, I just started thinking about potential. What would games with biology look like? For some reason that triggered a thought there. If we did learn all these ways of interacting with biology, could we also kind of gamify it in a way and generate a new interaction with biology?
Yeah, so one of my companies, Molecular Reality, is gamifying nanopores. So this is a way of interacting with biology, measuring the sizes of small organisms and things like that. So
talk to us more about it, please.
I mean, just I think the idea is making a consumer scientific product that's even simpler than a microscope, simpler than most scientific instruments that anybody can have, that tells you basically the sizes of things. It's based on the Coulter counter and the nanopore. It's kind of somewhere in between those two, and maybe even including those two, where you can measure sizes of large populations of things. For example, you might want to do that for microplastics, to see what the size distribution of microplastics might be as pollutants, or whether your microorganism aggregating or not, or whether big things are eating small things. So that can be that is being gamified, and the reason I brought that up was that it is already happening. But almost anything could be. To some extent, the iGEM competition is kind of a game where everybody's trying to make something that could be useful for society using parts that they inherited from previous years' competitions. They're not trying to do the equivalent of robot wars where they destroy each other's creations. They're trying to build. Yeah, well, that was intentionally. I think that was one of the more intelligent things we did as a community in the Sinberg community, as well as the iGEM community, is emphasizing proper human practices and ethical behavior. And it seems to be working. I mean, it seems like that's the culture that's growing up, rather than the black hat hacker or
yeah. And I think they've also developed like human practices. It's very, very central actually to iGEM. And I'd say that they still, so they've also introduced like a rewarding system if you do consider security, or you put security measures in place within whatever project you end up developing. So it's brilliant.
Yeah.
Sorry, just before we stop with the gamification, what do you mean, like, because I also recently learned about this expandable, what's it called? Expandable microscopy? Is that relevant at all? Okay.
Yeah, so expansion microscopy is something that Ed Boyden's group pioneered, and we've co-authored some papers with him on it. It's based on the principle that certain hydrogels will expand if you remove, let's say, counterions that are shielding the like charges. Like charges like to repel if they're not shielded by counterions. And a classic example of this is polyacrylate, which is present in diapers, which will expand, hundreds-fold when your baby releases their urine. So it's the same thing here. Typically, it's hard to get more than a 10, maybe 20-fold expansion but you can do multiple cycles and get bigger expansion. And that's linear dimensions. So the volume is the cube of that, whatever it is. So it allows you to do higher resolution microscopy without a fancier microscope. So you can use the simplest microscope to do this. And then the reverse is implosion, and that allows you to take something, manufacture something big and shrink it down. Yeah.
But yeah. Does it work with living organisms? Can you shrink and kind of magnify whilst keeping the
it would be a very special organism. You could do that. But I make a rule of never saying never or impossible, but
I like that.
It would be you might be able to do it with a quote multicellular organism where the cells don't expand, but the distance between the cells grows. So in other words, the cells are kept intact, but the hydrogel increases the distance between them. I'm not sure exactly why you would do that, but that would be one way to do it. You could think of like colonial multicellular, as opposed to highly developmentally constrained ones. Nevertheless, you could conceivably take a living thing, expand it, change it, shrink it back down, and it might function. But that hasn't been done. Yeah.
It hasn't been, because then that's you can really think about true meaning of like livables there, like if you and then literally interacting with a biological organism that's like either on you for a certain function, and then you kind of magnify it and tweak it and it has a different function, or I
think we're going to increasingly be hacking our inside and outside of our essentially our epithelium. So in our gut, there are already some companies that are producing microorganisms that interact with, that are in equilibrium with your blood, and hence in equilibrium with your whole body through the gastrointestinal cell lining. So, for example, there's some that work on the gut-brain axis. It's like gamma-aminobutyric acid. GABA is a neurotransmitter that is somewhat regulated. Its levels are somewhat regulated by your microorganisms. And then there are companies that are making bacteria that will detoxify. So like Fit Biomics will take lactic acid that builds up in athletic performance and turn it into, which is fairly toxic, and limits your final performance, and it'll turn into propionic acid, which is a much more positive.
And is this as a probiotic, or?
So as a probiotic, so for example, yogurt or what have you, and Synthorix does it for rare metabolic disorders, where you take the toxic buildup product and and take it to the step that's missing in the human, that present in every other human, but instead now it's in their gut rather than in their body proper. So there are a few examples like that.
Speaking about microbiome now, actually, you had one of the several projects that you had was the weather map, which had a beautiful vision of a weather map like collective, right? Monitoring or tracking kind of global microbiomes. Do you want to talk to us a little bit more about that and whether it's still going on or how it was initially set up?
Well, it was initially set up around 2003. At the time, there was no particular pandemic, I don't think, that I was responding to, but I was maybe anticipating one that happened a few years later. But the idea is that you take existing equipment and, and develop some new ones as well, so that everybody has their own little weather station. Weather is one of the best examples of citizen science. It's something that everybody feels they can understand, at least part of it. They worry about it almost every day. They talk to people about it. It's like one of the big icebreakers in conversations, talking about the weather.
Especially in the UK.
Yeah, exactly.
You can spend 10 minutes talking about it.
Right. And so we're getting to the point where nanopores might be generally available. Not the nanopores I was just talking about a minute ago, but actually DNA sequencing devices that they're in the order of smaller than a cell phone, maybe attached to your cell phone, and you could use that for reading out your air, your food, your mouth. And each individual reading might not be that interesting, but when you combine it as a community, if everybody shares that information, then you can see patient zero spreading, things spreading out from them. You can see, which seasonal flu is the one you should actually be paying attention to, rather than the ones that were anointed for the seasonal flu vaccine, et cetera. I mean, you can see whether a particular room is suitable for you, because you know what vaccines you've had, or what individual personalized medicine you need, allergies and so forth. And so by kind of like a community electronic network, this is cell phone mediated, like so much of our information is shared, but this would have some biological component. We do it a little bit already by monitoring sewage and monitoring. A lot of this is post-COVID type stuff, but also monitoring the queries that people are making on the internet. But imagine your phone actually was measuring the DNA in your environment, and that was multiplied by everybody on the planet doing it. That would be an incredibly powerful public health thing, and it could pay for itself, but because just like people are paying for weather, there's all kinds of weather apps and weather ads and so forth. Whether bioweather should be at least as interesting as weather should be. They're both could potentially save your life. If you slip and fall on the ice because you didn't know it was going to be icy, it's just as same kind of logic goes into avoiding avoidable public health infectious diseases.
Absolutely, and it's really bringing individualized or personalized medicine into realization.
Right. Way more affordable than, say, gene therapy, which I also do, but I am also quite critical of it in its most expensive form. I think there are alternatives, but we should always be looking for these win-win alternatives where you can have almost zero cost public health improvements.
And so with this the zero cost one, is this the aim with nanopores, the portable devices? Is that in the horizon?
Yeah, absolutely. I mean, it's a bit of a chicken and egg, but in a good way, in that as the market grows, the price reduces and the market grows more, and it keeps going like that. That's what happened. We helped DNA sequencing take that, and synthesis take that route. They're both about 20 million times cheaper than when we started. and the same thing I think could happen for consumer level devices of various sorts. The more you use it, more people use it, the quicker it becomes publicly available.
And talk to us a little bit about what happens to the data. I know you've already thought about privacy and security implications. Both, I mean, just a couple examples is like Nebula Genomics and HLTH network. Are you happy talking a little bit about these? And
yeah, sure.
Yeah.
So yeah, Nebula and the health network are using relatively new tools. I mean, there were ideas that go way back, but relatively new that they actually practically work, having to do with keeping records on the public blockchains. So you've got these ledgers that where it's hard to fake because they're interspersed with other information, and homomorphic encryption queries where you can ask a question about an encrypted data set without decrypting it. So and also by pooling data where you I mean I've been publicly critical of attempts to de-identify by pooling, but nevertheless there is some value to that, especially if you combine it with the blockchain and the homomorphic encryption queries. But the combination of these three things allow you to just like have this big feed of information that has geographical indicators. And of course, geography is very identifying, right? Especially if the resolution is very high, which it should be to be effective. And so I think efforts to de-identify are not as valuable as efforts to educate people and to destigmatize and to penalize people that are abusing the information. Rather than trying to protect the information, you aim for the consequences that you want, which is that people shouldn't use it for discrimination, and so forth. And there is a Genetic Information Non-Discrimination Act in the United States, but it's not universal throughout the world, and there's no guarantee that it can't reverse. So we need to bolster up that way of thinking, is that don't make everybody hide, make it so that it's safe to come out, which is kind of what's happened with gender identity and cancer, alcoholism. These things were all stigmatized, and now people talk about them more freely. That's the direction we should move in, rather than getting much better at hiding things, because I think that hiding things is where we get into trouble.
And you yourself have shared your genetic information, and in fact, at some point you were going to publish it as an NFT. Am I correct?
Right. That was more about education. But yes, not only my genome, but my medical records are publicly available, and I think that's I've had people volunteering who want to make it publicly available. So in case they're traveling somewhere, they don't have to get two hospitals that don't want to share data to share data. They can just download it from the internet. That's an example, but probably more importantly is the medical research community. It needs to be able to share data more fluidly without having to have complex squared institutions agreeing to the same thing. You just say, I'm downloading the data, right? That's it. I don't need to have a long negotiation with 500 other organizations.
I definitely agree with the perspective of security through obscurity as weak security, which you're alluding to. And I think this is the best way of explaining, or is it like stuff like zero knowledge approaches to sharing data is, where's Wally? Right? The game, you have a page instead, and you let someone know where Wally is without literally telling him where Wally is. You just cut out where he is on a page, and you give that page to someone. Is that like a fair layman's description of the techniques that we can use in securing and sharing data without actually sharing the data?
I think that still falls under the category of security by obscurity. You can still reconstruct where Wally is from that information. You can actually, where he actually is. Homomorphic encryption queries is more like I take my disk drive and I encrypt it and I hand it to you, and then you want to know, something personal in there, like my birthday. I give you the tools to find a whole variety of things like that in a one-off way, and you can find it without ever having, if you decrypt the disk drive, obviously you can find it, but you don't want to do that because for that brief period of time it's decrypted. Even if you immediately erase that copy, for that brief period of time, it's available for hacking or copying and so forth. So you need to keep it encrypted, and the homomorphic encryption query allows that to happen. I don't know what the perfect metaphor is, but it's.
Yeah, I was just gonna ask actually.
Yeah,
So you preempted that.
Yeah, it's. Yeah. It'd be something like you have a bunch of objects in a black bag, and you have a robot hand go in there and ask you if there's a rubber ball in there, right? And you don't actually know everything that's in there. The robot will only report whether it finds a rubber ball or not, or maybe even pulls it out and gives it to you.
Yeah.
But anyway, I think there's a growing number of tools like that are better. But even better still is just say, why are we hiding this? Why do we need homomorphic encryption? Because clearly nothing bad has happened to me in the 18 years since I first started putting my information online, my medical information. Now that's of course an anecdote and doesn't apply to everybody, but it's an open question: is if something bad could happen. Let's say I had some neurodegenerative disease, and my employer, Harvard, decided to fire me because I couldn't do my job in advance. That would be discrimination. They might or might not be justified. But the point is, there should be a law that says they can't do that, rather than a law that says, I have to keep all my information in this hyper secure vault. And it's not the fault. If somebody hacks into my hyper secure vault, that's unfortunate, rather than having consequences for that, which there often are.
Do you see any kind of cyber physical or like any security systems we can use actual biology for?
Yes and no. As I have one of my students is preparing a paper on encryption that is biochemically based, biologically based. I shouldn't give too many details about it, but you could add you encrypt into zeros and one. You encrypt your zeros and ones by classic, digital encryption, and then you can encrypt it further with this biological trick. There are a lot of it, has to do with practicality. It's like it depends at every stage in encryption is dependent upon what size key you need is dependent upon the compute power at the time, and it also depends on how well quantum computers, once they started arriving, how they might allow you to do things that weren't previously feasible. And the same thing's true for the biological encryption. It's dependent upon your ability to, say, sequence a lot of DNA or make copies and things like that. So almost all of these things have some way of hacking them if you have infinite compute power or infinite, sequencing power.
With that example of your student, again, if you can't say much, then that's fine. Is it working to protect against what was Peter Ney's, I think, work in biomalware, where he showed that he could store like a computer exploit into physical DNA that when sequenced compromised the computer and gave remote access to the malicious actor? Is that the, or well, what's the threat model for the
Well, it's just any data. It doesn't. Yeah, I mean that's a very specific example. Any data. So there is this whole field that we haven't talked about too much of storing digital information in biological materials, either DNA or long protein polymers, that sort of thing. And that most of the effort, I would say, there's an international consortium now of data storage in DNA and other bio materials. It's mostly focused on storing conventional digital data. Again, most revolutions, the first inclination is to apply it to things that we already do, when you really should be asking, what new things can you do? But and one of the new things you can do is you encode biological information in biological storage. So, for example, we've made a mouse that stores developmental data in every cell of its body, every nucleated cell in its body, for every stage from eggs to adult. And you can go in and randomly access any part of the body and ask what its journey looked like. And you can not only store developmental data, but also physiological data, like levels of hypoxic compounds. In principle, any physiological data that you can interface with a DNA writing device, anything where you have a sensor. And there's all, as far as I can tell, there's almost no limit to what you can make sensors for that are interfaced to DNA writing. And so we calculated that the amount of information stored in this mouse was just a little less than a terabyte of information, and it's stored in a nanogram of material. So it's 1/1000th of the mass of the mouse is dedicated to a terabyte drive. Now, I don't know about your terabyte drives, but mine are not in the nanogram range, the ones. And we're getting ready to scale that up to exabyte scale. So we think we could, if we use the so-called junk DNA of the genome, we've done some preliminary, we published some preliminary experiments where we've engineered junk DNA in human stem cells, and that looks like you could store exabyte of information rather than sub-terabyte in a mouse. Again, using a tiny fraction of its mass to do that. Maybe instead of nanograms, maybe micrograms. But out of a 30-gram mouse, that's a tiny tax to pay for storing this vast amount of information. And again, exabyte drives don't really even exist. You have to have like a set of drives that give you an exabyte, 10 to the 18th bytes of information. So in a way, that's protected simply by the inaccessibility. We don't really have a good way of reading an exabyte of biological information. So in a way, it's encrypted in a form that only later generations will be able to read the whole thing. But in the meantime, we can read pieces of it, just like in flight recorders. If you think of the worldwide set of flight recorders, it's a vast amount of information, most of which is never read. As far as I know, most flights go fine, and so that you just write over the previous information in the next flight. And so it's an example where you write a lot and read a little, and that's very valuable. So you read, if something happens to a particular part of your body, then you read just that part in just the period of time that you're interested in.
So my mind is blown, I think, a little bit. So wait, so you're storing in each physiological information to do what? So what could you theoretically, if you didn't have the issue of reading or the limitation, what could you potentially do?
Well, it's not just potentially, this is what we have done.
Yeah, what you have done then.
and it's quite a field at this point.
Never use potential with George. It's what we are doing.
So for example, we've recorded the decision tree, the developmental biology decision tree that says, these two cells were related to a parent cell, and one of them decided to become erythrocytes, and the other one decided to become lymphocytes, right? And that information is stored in the final cells that exist at any time when you pluck them out and sequence them. It'll tell you, you can reconstruct a tree of information that says what happened at every cell division all the way. Similarly, for physiology, you can have a compound for which you have a DNA sensor, which then either you write into DNA either by a polymerase action or by a recombination action or editing. It's kind of DNA editing's in the news. So any of those, if it's interfaced with a active site allosteric site that is responsive to a particular chemical, then you record the levels of that chemical as the levels of editing. So you can turn a time series of physiology into a linear mapping on this digital recorder, which is DNA.
Which is insane.
Yeah, it is.
Just by the by, yeah.
It's even more insane when you think of it. Everything that we see about our world, what we hear, see, taste, touch, all those senses are physiology, right? So, when we touch something, there's a little set of neurochemical reactions that are going on. You could record those in the tip of your finger, and it'll stay there, and you know what part of your body you're recording because of location. You know the timing because where it is along the DNA, and you know the amount. And so, in principle, if you think of a retina, each of those cells has this physiology, and you can record in that cell. And so you can actually record videos in the eye of an insect, right? It can say, this is what the insect saw at this moment. And so you have essentially a digital camcorder that's entirely biological.
Is insane. So those dreams of days in the future, again, can't use in the future with you anymore, but when we want to record using our own eyes, no devices, no cameras, no, GoPro is the closest thing, but you still have to attach it, and it doesn't really get your worldview. Yeah, it is here. It is in the making.
But yeah, if you think of drones are fairly noisy, even the smallest ones. But there are all kinds of flying insects. You don't even know they're there. There's some of them that are sub-millimeter in size. They're totally quiet. And even birds. I mean, I've had birds fly past my ear, and the only time I hear them is during the fraction of a second they're actually next to my ear. And actually, I think it's just as likely that's the air flapping. I'm feeling the air rather than I'm actually hearing the sound. I mean, they're very quiet. So these are way better than drones.
No, absolutely. I really like the when you mentioned the senses, or recording the senses, and then I don't know, we kind of went down that rabbit hole last time we spoke as well. Whenever I hear the word senses, I just go down the road of synesthesia, and now I'm thinking usual story, this, George. My mom has synesthesia, and I'm completely envious because it's supposed to be genetic, and it obviously skipped a generation. And there's hopes of gene therapies to induce synesthesia, but wait a minute, here we're talking about, does that mean we can create a meta senses type thing? If we can record these sensations, could we then mix and match and create meta senses? And
well,
then
absolutely. We're essentially doing synesthesia, and we're converting the sensing of photons into the sensing of DNA at the DNA level, or sense the mechanotransducers that occur in your inner ear for sound and balance get changed into DNA, and then you can mix these in any way you want as you read them out. Well, you probably, for now at least, read them into a computer first, and then the computer can make synthetic biology that does whatever you want. So yeah, I mean, we're well into the era where synesthesia is harnessable rather than merely something that you inherit randomly.
Awesome. Speaking of, so we mentioned gene therapy. Do you want to talk to us about the work that you're doing, and mainly just for our listeners, tell us what a supercell is, why is it important, and how it relates to the work that you're doing?
Right. So gene and cell therapy are kind of coupled in my mind, and we work on both. And uh, when I started working on them, just like when I started working on sequencing, I was not happy with the projected costs. The projected costs of sequencing a human genome were $3 billion for a low quality genome, and now it's 250 for a high quality, meaning both your mother and your father's inheritance. And the same thing for gene therapy. It was projected there would be a couple $1 million per dose, making the most expensive category of drug in history. And I didn't like that. So we'll get back to supercells in just a minute. But first, the cost of gene therapy is, and this relates to one of our previous topics, related to the market size. So the ones that cost $2-3 million a dose have incredibly tiny market size. So the fixed cost of research and development has to be balanced out by that tiny denominator. But if you get to big market, like the whole world, which happened for the pandemic, you got the top 5 vaccines were formulated as gene therapies. This isn't commonly the way they refer to it, but there's no doubt that they're full-on gene therapies. And the particular one that was an adenoviral capsid around the double-stranded DNA representing the COVID spike protein, that got as low as $2 a dose. So without losing money. So that's hopeful, I think, for gene therapy. Now, cell therapies, you have kind of 2 forks, neither of which is quite as easy as gene therapy, because one fork is autologous, meaning cells from your own body. And that means that you have to do these really so-called good manufacturing principles for each person. So you have to start up a whole factory for that person. And then the other way is making a generic cell therapy, which works on everybody or large sectors of society. That one probably is more cost effective, but there's a lot more R&D to do to figure out how to not get it rejected and so forth. Now, supercells that you asked about are the notion that if you're replacing, let's say you're doing a transplant, you need to replace something that's broken, or you're putting something, a cell therapy in, you don't want it to succumb to things that the thing you're replacing. So, for example, you might be replacing a liver due to hepatitis or cirrhosis. So you'd like your new incoming liver to be alcohol resistant and Hep ABC resistant. And then you might want them to be enhanced in various other ways. So enhancement is something that we don't like to talk about if it's about germline human enhancement or super soldiers. But if it's just trying to make regular medicine work well, so for example, so you want to overcome the normal rejection, immune rejection of transplants. So that's a kind of an enhancement. You're making something that's more of a universal donor, which we think of with blood as O as the universal blood donor, but the equivalent for tissues is more complicated, but it is totally feasible. And we're doing it in our transplants from pigs to humans. So immune resistant, pathogen resistant, possibly cryopreservable, so cold resistant, resistant to senescence and cancer. Those are kind of like the 2 things that almost everybody dies of. About 13% for cancer and about 90% for other age-related diseases. So you don't want your cell therapy or transplant to easily succumb to that if you know how to avoid it. And we do know how to reverse and protect aging in animals. So it's just a matter of adapting those for cell therapies.
Now I understand its name, supercell.
Exactly. So one of the first supercells we made was for neurodegenerative diseases that are due to autoimmunity, like multiple sclerosis. We did this in a mouse model, putting in human cell therapy into the mouse brain, and they were resistant to the autoimmune reaction because they would produce their own cytokines that would kind of manipulate the incoming hazardous environment. But there are many other things that I kind of alluded to that you can do to make supercells.
Awesome. I'm a little wary of time, but I'm gonna ask anyway. Do you mind talking to us a little bit about what a functional connectome is as well? This is another area of your research.
Well, so the connectome, let's start with that, is how your cells in your body touch other cells. Now, for some cells, that's not the first thing you think about. You think of, like, lymphocytes floating around, and they might interact with a bacterium, okay? But they actually interact with all kinds of epithelial molecules and other immune molecules in the immune cells. But the poster child for all connectomes is the nervous system, and there you have thousands of connections between your favorite cell you're looking at and other cells. And there's a little redundancy, but these are important connections, and you can't just say, oh, I'm gonna, I might replace. We were talking about replacing the liver that was damaged, or replacing oligodendrocytes that will remyelinate, demyelination in multiple sclerosis. A lot of parts of the brain are not that easy to replace. Maybe Parkinson's, maybe multiple sclerosis, but a lot of other things, you need to keep those connections. So anyway, first you need to be able to analyze those connections. It's typically done now by serial sectioning with either fluorescence or electron microscopy, but that's obviously destructive. And also, it only tells you what the wiring diagram, doesn't tell you how it's being used. So the functional connectome, or as we call the brain activity map, which is sort of the basis of our proposal that started the brain initiative that Obama announced back in 2012, I think. You want to know how the information flows. So that's the functional connectome. You want to know what the neurotransmitters are, their neuromodulatory molecules, peptides, and so forth. You want to know that, map that out. You want to know the direction it's going, presynaptic to postsynaptic. Then there's another aspect of so once you get the ability to do that, ideally do that in a non-invasive way, so you don't have to serial section through my brain while I'm alive.
How do you do it?
Well, I mean, we don't know how you do it yet. We barely know how to do it with serial sectioning, but it would have something to do with the fact that there's traffic of cells across the blood-brain barrier that can kind of move around and sense the core, like, hub of the neurons, as well as their synapses, and you can and so your sensory cell. We talked just a little bit ago about how cells can record their life experience, or part of it, in DNA. In principle, you could have little eavesdropping cells that go all over the brain and record what's going on, either in a targeted way or in a generic way. And then you have to have things that act upon that, ideally without going through a computer. So it's just keep it all in the
biosphere.
Engineering, yeah. So then the next, so challenge number one is go from serial section connectomics to functional connectomes in a living organism. And then the next step up from that is anything you can read, you can write. That's my research motto. And so then is, how do you write a connectome? And that gets back to the earlier topic of how do we learn the language of developmental biology as a 3D printing mechanism. And we're not limited. When you read the lab mission statement, you said we're not limited by regular biology. You can be inspired by biology, but we're free to use other tricks to make a synthetic connectome. So making an arrangement where you can either reproducibly or one-off make a particular set of interconnections among neurons, so you can study them, so you can test drugs, so you can make modules that are transplantable, and so on.
I'm as usual left speechless after talking to you. How do you? Because we've said it now at least a couple of times. How do you, especially with your students in your research group, how do you enforce that kind of uh, people usually say thinking outside the box, or how do you bypass certain paradigms that we have or certain ways of thinking, even with like I was listening to a podcast the other day actually that was a person trying to explain what thinking means, but couldn't do it without using computer language or like the words processing or bandwidth, etc. So how do you implement that? How do you enforce that? This kind of more out of the box thinking to try and find new ways or like you mentioned in this example with the synthetic connectome, bypassing kind of the paradigms of how we usually think of 3D printing, right? Yeah, I'd love to hear your thoughts on that.
Yeah, so there are a few things. There are probably some things that I don't even know we're doing. Because with evolution is with engineered evolution is a combination of random things and engineered things. So the engineered part of it, social engineering, is you first of all create a culture where failure is embraced. Again, it's this needle in the haystack. If you think that is all hay, then it's impossible to find a needle there. But if you think there's a needle in there and you can calculate roughly how many, strands of hay you have to go through, then you will find it. So that's one component of out of the box. There's also a positive feedback loop that once they've seen a few out of the box things work, then they have more trust, more faith, and they're willing to take a bigger jump. Now, you also have to create an environment where even though failure is an option and where there's this positive feedback loop where they're willing to take bigger jumps, you need to have a culture where you protect people. So that means that as they're taking this, they're going for their passion that may seem impossible to the rest of the world, but we have a way of de-risking. We have a way of evaluating things that look impossible that are actually low-hanging fruit in some cases. But the other way of de-risking it is to give them two projects or let them pick two projects, one of which is a surer thing. But the way most biologists think of a sure thing is they dial back their expectations. They dial down to a mediocre project. Instead, what we do is we take a sure thing in our group is something that was crazy and has slowly worked its way up to publishable. And now it becomes, it's not mediocre because it passed through this daunting history, but people can have that as their second project. Maybe they'd be middle author on a paper, but still it's a sure thing without being mediocre. So that combination of things I think helps the thinking out of the box. The final thing, not the final thing, but the final thing I'll mention is that if you want to build an out of the box team, it's hard to build it out of disciplinarians. If you want an interdisciplinary team, it's hard to build it out of a summation of disciplines. And I think this is a mistake that a lot of, heads of state and companies and universities make, is they figure we put a big pot of gold in the middle of the table and invite, physicists and language experts and biologists to join in. They basically will divide up the pot and go out, run away from each other. Instead, if you take people that are multilingual, polyglots and polymaths, and even if they don't happen to share a discipline or a language, they have proven that they are motivated to learn multiple languages, and they will find a common language, and then you can stitch together this network of expertise. So I think that process of taking interdisciplinary people and making interdisciplinary teams from it, even if your goal wasn't out of the box, you will achieve out of the box that way. And then I guess one last thing is people who have had the misfortune of being different, let's say, growing up chubby or red hair or, high functioning autistic or, you name it, almost anything that makes you out of the middle of the bell curve. you get made fun of. You don't know you don't know how to compensate. That actually turns into an advantage in adult life, especially in the part of society that embraces out of the box thinking, because it doesn't matter. I mean, you don't have to have like the genius gene. You just have to have been different and ostracized at some point in your life. And it has you have a slight edge, no guarantee that you'll be able to think out of the box, just because you've had to your whole life just to cope.
I'm curious to hear, George, if you or what a lot of people consider like the research that you do or radical or crazy, but I want to flip it around and ask from your perspective, because you alluded to that kind of calibration almost of like what is considered a radical research project from your perspective, which for someone else would be like super duper radical. But what's the most radical idea that you've heard in your field, and what do you think about it?
Well, my fields, plural,
Yeah.
Most of them come from my fantasies, not from the internet or from books. I mean, science fiction, maybe, but usually science fiction has a problem. They haven't really connected the dots well enough that you could that you can assess whether it's a cool idea or not. But I would say one of the most one that inspired me to take it a little further was the idea of making small probes, space probes. So there was this breakthrough Starshot thing that aimed for scaling down our space probes from kilogram size, multi-kilogram size down to gram size, and then you could do more of them. And I said, reductio ad absurdum, why don't we scale it down to picogram size? So kilogram to gram is 1,000-fold, gram to picogram is 10 to the 12th fold. And then because the synthetic biology, I thought, well, picogram is actually pretty big. It's it's big enough for a cell that can replicate, and then it can become kilogram size. So basically, it's a way of packing a kilogram into a picogram, sending it at near speed of light via light sails and whatnot at a billionth the cost. So I proposed that in a paper that just came out in astrobiology, where I went through some of the things we've been talking about today, the ability of synthetic biology to engineer inorganic materials as well as organic, and then hence possibly being able to make a communication device, that interstellar communication. The problem with interstellar communication right now is you can't, there's nothing at the other end. So you can send something out, but you don't know what. But if you send, if you
left hanging, literally.
Yeah, exactly. It's like high five. Wait a minute. 10 to the 4th years later. But if you send this picogram probe out and it expands and uses its developmental program to make a communication device, which shouldn't be that much harder than programming into DNA how to make birds' nests and all the wonderful things that are made by biology. So yeah, that's a wild idea.
Pretty wild, yeah. I was gonna ask like what's in the horizon, but totally covered that. But okay, let's step into like 2073. But I think we covered that too. I usually just ask you to give me like 10 words to describe society in 50 years from now.
Yeah.
But do you feel like you just have?
No. There's much more, but it's also it's getting harder and harder because of the exponential that we're on. We're getting exponentially more capable, lower costs, higher quality, more diversity of technologies and materials and so forth. So I think we may be unrecognizable in 50 years. Now, there probably will always be some residual that is recognizable, and we'll probably communicate with the unrecognizable via the intermediates. But yeah, I mean, it's not impossible that we are that we have a different form and function, and we might be an interstellar species by then. I would hope so. Certainly, we will have occupied a huge fraction of our solar system, and that will make us much safer as a species. And we can take whatever species we want with us, kind of a Noah's Ark thing.
Yeah.
A little known fact is there's about 100,000 times more liquid water in our solar system, liquid, not frozen, liquid water in our solar system than there is on Earth. Earth is a tiny droplet of water compared to the rest of the solar system.
I did not know that.
Yeah.
I did not know that at all.
Yeah. Even some of my astronomer and astrophysics friends were a little surprised when I told them that. Yeah.
That is insane. I mean, well,
you can look it up. It's there.
Yeah,
Now, a lot of it's hard to get at because it's got a frozen layer on top of it, but still, it's there. And that vast amount of water, since water is the key component of life, every kind of life that we know of so far, that amount of water could have living things in it, replicating things that completely eclipse the kind of diversity we have on Earth. I'm not saying there's anything living anywhere, but if there is, there could be a fifth more of them.
And we still don't know what's the percentage now. We don't even know all the living creatures in our own droplet. I was going to say deep blue giant sea droplet of sea in comparison to our solar system.
Right. So, yeah. Well, although we're getting there, we're on a path to enumerating all the big organisms, and even on a path towards enumerating the microbial and viral parts of our biosphere. There's almost, for better or for worse, there's almost no part of the biosphere that we haven't touched our toe into, at least. So
just a little bit.
Yeah, it's not. I mean, the bottom of the ocean is probably the least explored, but we have sent several people and devices all the way down to the bottom of the Marianas Trench. So we just haven't done that for every square centimeter in the ocean. But I wouldn't be surprised if every square centimeter of land has been touched at least once by a human foot or shoe. Yeah.
Okay, so unrecognizable, interstellar, safer. I'm describing the future in 10 days. I'm just helping you count.
Oh, yeah.
Different form and function. We have 5 more.
Yeah.
I'm pressing on you a little bit.
Probably, hopefully, very caring and intelligent.
Caring and intelligent. Yeah. I like that. 3 more.
Well, I think safety can be broken up into various components. Safe from ourselves, safe from global catastrophic risks that we just can't protect ourselves from, other than being diaspora. I don't know how you count those words, but
yeah, I was gonna say,
I'm clearly playing it that slightly different than your play.
Awesome. Thank you, George. And one last thing, knowing that we have interesting guests on this podcast, what question would you ask our next guest?
I think the kind of questions you asked are spot on. I think very few people would answer them the way I did, but they're still good questions.
Specifically, though, is there something that you want to ask? Either whoever the guest.
I think if I want to ask it, I ask it. I don't have a long-term plan for asking questions. It's all short-term. I have long-term plans for other things, but asking questions, it's an instant gratification thing, especially with the internet and the fact that people do answer my emails.
Absolutely.
Yeah, but you're asking good questions, and keep up the good fight there.
Perfect. Thank you so much. And with that, just remember, guys, never ask future questions to George Church because he is doing the research. It's in the present tense. Thank you so much, George, for being on. And we're going to have a second one because I think we literally just scratched the surface.
Okay, great.